Thank you, Gauri, for briefing about the PG diploma course. So, one thing, have you noticed that when she was discussing about the topics, so we heard a lot about omics. So, what are omics? So when we are so here, as you likely know that the central dogma of biology, it details how information is encoded in an organism and it eventually becomes phenotype. So here what happens is the crucial information is encoded in a cell's genes and that is transcribed into RNA, which is then translated into protein. So here, of course, the regulation of how DNA is transcribed is often controlled by, we can say, non-genetic regulatory mechanisms, such as epigenomic or epigenetic regulation. Here, when we're talking about omics, so each layer of the central dogma, the genome, the epigenome, the transcriptome, and the proteome are considered as omes. And omics is an umbrella, we can say umbrella term, referring to the study of individual or multiple levels of central dogma. So, yeah. So now, when we are talking about genomics or when you're talking about genome, it is the complete description of one's genetic makeup. And we can say the variation in the genes defines the phenotype that can underline many diseases or different traits we can discuss. So such variation could either be germline variation or somatic variation. And studying these kind of variation with different approaches such as whole genome sequencing, whole exome sequencing, or targeted sequencing can come under genomics. So whereas when we are talking about transcriptomics, so here not all of the changes to the genome are due to changes in base pairings. So what you can say that the way in which DNA is organized or chemically modified also significantly tells us the story of gene influencing phenotypes. Yeah. So this is about epigenomics, what I'm talking about. So here, the cumulative term studying for non-genetic changes is called epigenomics. So on the other hand, RNAs are the functional output of one genes. So your all cells, we can say that have the same DNA, yet the gene expression varies drastically between different cell types. And studying these gene expression comes under transcriptomics. So at the last layer or level, we can say, is the proteome. So it is the ohm that is closest to the phenotype. It is the functional output of all other levels of cellular regulation, we can say, including the genome, the epigenome, and transcriptome. And this is where researchers often look when they're interested in finding things like biomarkers. So, like the question comes that why do we need to study omics? So, if we omics or multiomics, so if we take cancer research as an example, here each ohm tells us something distinct story about cancer biology. If you're looking at the genome, it often provides the mutational profiles that help to predict patient diagnostic, patient prognosis, or it can also classify tumors into meaningful groups, or even we can say diagnose cancers. So whereas when you are considering the transcriptome here, since it is the functional output of one's, we can say gene, it can show us pathways that are up or we can say down regulating in specific cancer. Whereas the epigenome, it adds an explanatory, we can say mechanistic dimension to RNA expression. Here, it can further refine tumor classification. And finally, the proteome, which is the closest to the cellular phenotype, it can provide biomarkers and other phenotypic information that responds to all, or we can say, predict the therapeutic responses. So this is how we should integrate our further research using omics. But like all other technologies, so even we can say omics is also plagued with different challenges. So what are the challenges? So biological complexities is one thing. There are ethical and privacy concerns. There is a rapid evolution or in technology, because technology is dynamic, right? There are wet lab challenges, and these are the challenges we need to address. but how can we address them? So we can address them by reshaping the skill-based approach. So this is what actually we are doing now by this PG diploma course. So we are designing a training program that will help to build a skilled workforce which is proficient in omics technologies, what we have discussed. It can narrow down the training and expertise gap. It can provide skill sets to efficiently plan multi-omics experiments. Yeah, but the thing what actually we believe is, so whatever thing the theory what we have learned, so based on that we can say that the true development of skill comes from hands-on training, where practical application turns the theoretical knowledge into real-world expertise. And this is what we believe when we are delivering the PG diploma course to you all. So whatever theory we have learned or you will learn in the course, we will do a hands-on training. So we will try to apply with the real-world samples. And this is what actually we did in campus immersion a while back. So, and actually with this, what I'll do, I'll hand over to Dr. Mohit, where he will brief you about the data analytics part of the course.